Okay, so this is the second part of the test review. So we went through passive transport, active transport, enzymes, the difference between prokaryote and eukaryote. Endosymbiosis is when something's taken into a cell. Exos when it's taken out. Cho-cho-chon-chomp, the difference between cells and viruses. That was active transport. And we talked about the phospholipid bilayer and how you spot that. And the job of the cell membrane is to maintain homeostasis by being the connection between the outside world of the cell and the inside world of the cell. So let's have a quick look at these ones. Okay, this is kind of a random thing, but here is obviously a plant, and we know that plant roots, one of the primary functions of a plant root system is to take water into the cell, into the root cells, and then those get transferred up into the leaves. We know that plants need oxygen, and we know plants need carbon dioxide, or they take in carbon dioxide when they do photosynthesis, and they release oxygen as a product of photosynthesis, and then cell respiration takes in oxygen and releases carbon dioxide. So gases get into and out of a plant in the leaves, right? That's how gases, oxygen and carbon dioxide, go in and out of the plant. But water only goes in through the roots. Okay. All right. Diffusion, facilitated diffusion, and osmosis. Let's talk about these three words. Are all similar, but they all have slight differences. All of them, diffusion, facilitated diffusion, and osmosis, are when a substance moves into or out of the cell by going down its concentration gradient, going from where there's more of it to where there's less of it, high concentration to low concentration. And because of that, because we are going down the concentration gradient, it is passive. It does not require energy. So diffusion is kind of a general term. You know, if you're in my room, that sometimes I turn a wax warmer on, and you walk in, you immediately can, you know, after a little while, you can smell whatever I'm melting to make the room smell good. That's because close to the wax warmer, you have lots of scent particles, and they spread out. They move from where there's more of it to where there's less of it. So anything can diffuse, right? So this is the spreading out or the passing into or out of a cell, right, of anything, right? Anything. Now, some things like really big molecules or polar molecules like water, they cannot get into or out of a cell on their own. They need some help. Facilitate means to help. And if you recall, you can always tell if it's facilitated diffusion because you've got the phospholipid bilayer, but you'll have a protein molecule, usually a channel protein, with kind of like a passageway going down through the middle, right? So if you see something going from where there's more of it to where there's less of it, from high to low, but it's passing through the protein molecule, then that's going to be facilitated diffusion. Facilitated diffusion, anything that has trouble going from high to low on its own can get helped by a protein molecule, and that's called facilitated diffusion. Osmosis is diffusion of H2O only. It's the special diffusion of water. It is where we have water moves from where there's more of it, from a high concentration of water, to where there's less of it, to a low concentration of water. Water always moves from more to less, and that's got its own special name. Remember, this is biology. use extra names whenever we can, and that's called osmosis. So osmosis, anytime you see the word osmosis in any test question, you are going to look for H2O in the answer. These two things always go together. All right, let's talk about hypotonic and hypertonic, because I know that this is a little bit of a, complicated, little complex idea. Hypotonic is where you've got lots of water outside of a cell and less water inside of a cell. The opposite, hypertonic, is where you've got more water inside the cell and less outside of the cell. We know, because we just talked about osmosis, osmosis is when water moves from where there's more of it to where there's less of it. So if you put a cell in pure water, you put it in a hypertonic environment, and water goes from where there's more of it to where there's less of it, and the cell will expand. Water goes in, right? It will swell, and then it has the ability to burst, because it will just get bigger and bigger and bigger, just like I said, like a balloon. And you put one puff of air too much in it, the whole thing bursts. Cells that are put in a hypertonic, that's a very watery environment, will get bigger. On the other hand, a hypertonic environment is one where there's very little water outside. So now we've got more water inside the cell than outside the cell. Water always moves by osmosis from where there's more of it to where there's less of it. So it's going to go out of the cell. And this time, instead of the cell getting bigger, it's going to shrink. It will shrink and shrink and shrink until it's completely just shriveled up, and that's called plasmolysis. And that's what happens just before the cell will absolutely destroy itself. That's called plasmolysis. So hypo is where you've got more water outside than inside. The cells expand, they take in water. Hypertonic is where you've got more water inside, and the cells will lose water and they will shrink. Okay, it's very important that you know the difference between those. So if I took a cell and put it in a hypotonic environment, I would expect it to get bigger. If I take a cell and put it in a hypertonic environment, it will get smaller. This is something that organisms that live in things like pond water have to deal with, because if you live in water and you're a single cell, you are constantly filling up with water. And so you have to be, a bit like, if your boat fills up with water, you have to get rid of the water. You have to pump it out constantly because it keeps coming in. Okay, so cells and organisms that live in pond water or aquatic environments, they have this problem that they're constantly filling up with water, and so they have to pump that water out, right? If you took a cell that lived in pure water and you put it in a salt solution, the cell itself would lose all the water and it would shrink because you're putting it in a hypertonic environment. All right, hormones. We talked a little bit about hormone. In fact, I'm going to come back to that one. We're going to talk about homeostasis. Homeostasis is how we maintain a steady, normal environment. A homeostatic response always starts with a stimulus, right? So if you remember, we talked about if you were controlling your blood glucose, the stimulus would be eating something that had a lot of carbohydrates in that got digested into glucose. That causes a part of your body to my lights just went off, so I'm going to have to reconfigure there just a little bit. Okay, that causes, whoops, everything's falling to pieces today. Okay, so if you eat a bunch of sugar, that causes your blood glucose level to go up. That is detected, right? It's detected. So in the case of blood sugar, your pancreas detects that glucose levels have increased, and that causes a response. In the response of high blood glucose, it would be producing insulin. Okay. If the stimulus was, you're working out on a really hot day, right? The stimulus would be an increase in your body temperature. That would be detected by the hypothalamus, which would have a response, which would be to make you sweat. Okay, that would be stimulus detection response. But it can also go in the opposite direction. So you could, for example, I was watching a program where cows had twins, and the mother had a stimulus of low calcium in the blood, and that was detected by a part of the brain, and the response was to start taking calcium away from the bones of the mother. And the reason she had low calcium was because both calves were nursing, and they were taking all her milk away, and she was making milk so fast that she had to get extra calcium, and that would cause a problem with, for example, muscles being able to move. So the stimulus can go down. So you could have been working out really hard and not eating anything, and that again would lead to a low stimulus of a low blood glucose, which would be detected by your pancreas, and this time the pancreas would produce a separate hormone, a different hormone called glucagon, which its job is to put extra glucose back into your blood, so you have a response. So you can see it can go either way. It can go up or it can go down. This is called where you have a stimulus which is detected and causes a response that brings whatever it is, glucose, calcium, the amount of water in your blood, the temperature. This is called negative feedback. All right, this is called a feedback loop. Okay, and feedback loops are how we maintain homeostasis. Now, going back over here to hormones. Okay, so hormones are chemical messengers. Hormones are also proteins. Okay, they are proteins which are produced in one part of the body, usually by some type of gland, and they travel in the bloodstream to somewhere where they make the response happen. And proteins are part of our endocrine system. Okay? So when you have high blood glucose, right, your digestion system, your endocrine system, they work together to control your blood glucose levels. Okay? So the endocrine system, the system that produces hormones, always works with another system, okay, to maintain homeostasis, to keep everything at normal levels. Okay, that is the review that goes with this test. If you have, I would strongly encourage you to review everything before the test, including the pre-test questions, and you should be pretty well prepared for your test then.